This is a production of Cornell University.
Good afternoon, everyone. As Jenny said, my name is Megan Williams, and today I will be discussing our work on investigating the nitrogen management and alfalfa variety performance in a dual-use Kernza alfalfa intercropping system. So let's start out by breaking this title down into its components to help us better understand the why and the how of this study, starting with perennial crops and their benefits. First, some of the stats. The agricultural sector accounts for more than 1/3 of all global greenhouse gas emissions, with around 11% of those emissions coming from synthetic nitrogen fertilizers, including its manufacturing, transportation, application, and post-application soil emissions, which makes synthetic nitrogen fertilizer account for around 2% of global annual greenhouse gas emissions. Synthetic nitrogen fertilizers are the main source of nitrogen in conventional agriculture, and wheat is one of its top users as one of the top 3 crops in the world, covering around 1.5% of the Earth's land area. Perennial grains have been identified as a potential sustainable alternative to annual grains like wheat due to their wide-ranging benefits, which include soil health benefits from increased below-ground biomass inputs, improved water infiltration and access, carbon sequestration, and lower overall carbon emissions, with their year-to-year longevity allowing for reduced tillage and for fewer tractor passes for planting. Here, we can see a side-by-side comparison of the root profiles of annual wheat here on the right, and a perennial grain crop known as intermediate wheatgrass here on the left. These deep roots are what are largely responsible for the improvements to soil health. Next, let's dive deeper into intermediate wheatgrass as a perennial grain crop, also known as Kernza. Just a disclaimer, Kernza is technically just the trade name for a specific cultivar of the grain product of intermediate wheatgrass, but because Kernza is a lot easier to say than intermediate wheatgrass, and because we did use that specific cultivar in our work, I'll just be saying Kernza for the rest of the presentation. So, while initially cultivated in the US as a forage crop, intermediate wheatgrass was selected for development as a perennial grain species over other perennial grasses for its traits that include high grain yield and size, as well as grain qualities like nutrition and taste. The intermediate wheatgrass cultivar, known as Minnesota Clearwater, became the first perennial grain commercially available under the trade name Kernza. Kernza grain has been used to make a wide range of products, including beer, flour, pasta, cereal, and more. And I personally can endorse the fact that it's very tasty. However, despite Kernza being selected for development as a perennial grain, there are still reasons why Kernza is not yet a household name. Despite successful breeding efforts to increase its grain size, Kernza grain is still about 1/3 the size of commercial annual wheat. It has been estimated that it'll take another 24 breeding cycles to get Kernza grain size up to par with wheat. Additionally, Kernza grain yields consistently do decline over time. In this graph from Locatelli et al., we see 2 different populations of intermediate wheatgrass, both significantly declining in grain yield from year 1 to year 2. This grain yield decline makes farmers less likely to want to maintain a Kernza stand for multiple years, thus negating some of the perennial benefits. So, beyond breeding for grain yield maintenance over time, research is being carried out to understand how agronomic management practices can affect Kernza grain yields over time, while still maintaining the sustainability benefits of its perenniality. Findings in the 2018 paper by Topkes et al. Suggest that intercropping Kernza with the legume forage crop does have the potential to better maintain Kernza grain yields over time when compared to fertilized monoculture alone. So, next we're going to talk about how intercropping sits in our sustainable perennial framework. Intercropping is the practice of having 2 or more species of crops in one field at the same time. Studies have shown that intercropping has a host of benefits compared to conventional monoculture, including yield increases, which can lead to a higher land use efficiency, where the same yields can be produced on less land, as well as higher yield stability over time. We can see these trends here in this figure from Lee et al., where they intercropped maize with other grain crops, including wheat, faba bean, soybean, chickpea, and oilseed. Across multiple sites and nitrogen rates, they found that grain yields were consistently higher in intercrop than in monoculture, and that average grain yield increased over time when compared to monoculture. Interspecies interactions lead to effects like weed suppression, increases in nitrogen and carbon in the topsoil, beneficial insect refuge, and disease and pest suppression. All of these effects and more come together to create the increases in yield, land use efficiency, and stability documented in intercrops. Now that we understand the benefits of perennials, kernza, and intercropping in general, let's look at why we specifically chose alfalfa to intercrop with our kernza. Alfalfa is a legume, and legumes symbiotically associate with nitrogen-fixing rhizobia bacteria in the soil, and by trading carbon resources for the nitrogen fixed by these rhizobia, they live in the nodules formed on the legume's roots, like you can see in the picture here on the right. And this nitrogen not only benefits the legume crop and reduces their need for synthetic nitrogen fertilization, but also can contribute nitrogen to the soils to be taken up by other plants in the intercropping system. Nitrogen transfer to soils and crops is accomplished through root and nodule tissue decomposition, root exudation, or even directly from the legume to nearby crops through plant-associated mycorrhizae-mediated transfer. Research has shown that legumes in intercrop with non-legumes do have the capacity to transfer as much nitrogen per area as monoculture legumes. And if enough nitrogen can be transferred from legumes to the other crop and intercrop to still support profitable crop production, then less synthetic nitrogen fertilizer can be applied overall to the intercrop, reducing both fertilizer costs and associated emissions. Alfalfa was selected for intercropping with kernza in the system because it is a widely used, profitable, and perennial forage crop. Studies have found that alfalfa nitrogen transfer to grasses like kernza increases over time with production year, indicating that its use in a perennial intercropping system can not only confer previously mentioned intercropping benefits, but also more nitrogen to the system as each year passes. As alfalfa is widely used, there are many varieties that have been bred for specific performance metrics. One metric is fall dormancy rating, which rates alfalfa fall regrowth on a scale of 1 to 11, with 1 having the least fall regrowth and 11 having the greatest fall regrowth. Fall dormancy is loosely correlated with winter hardiness, and fall dormancy ratings between 3 and 5 are well suited for northern states that experience soil freezing. And so we used ratings of 3 to 5 in our alfalfa varieties selected for this experiment. And here in this picture, we can see some alfalfa and some kernza in our crop together here at New York. So even if intercropping kernza with the legume is able to help slow grain yield decline and reduce nitrogen fertilizer costs, kernza grain still simply is not yet as high yielding and thus not as profitable when compared to annual wheat. So farmers interested in growing kernza may need to tailor their management techniques to maximize profit. Utilizing a dual-use system is one potential method. A dual-use cropping system is one with the intended purpose of both harvesting grain and utilizing vegetative growth as forage for livestock. Economic analyses of dual-use systems have shown that it can both improve the profitability of a cropping system and reduce business risk. Additionally, dual-use systems can provide a bridge in feed quality and quantity gaps for livestock at different times of the season. Analyses of kernza systems have specifically shown that the financial risk of kernza grain production can be reduced by managing the system for a high forage yield, thus providing the farmer income not only from grain, but from its forage as well. Further, intercropping alfalfa with kernza would then not only benefit farmer bottom lines by increasing yields per area and reducing nitrogen fertilizer costs, but then also presents the opportunity for multiple sources of income through the dual-use cropping system. That being said, though, ideal kernza management strategies to maximize profits are still largely dependent on kernza grain prices at market, which are still shifting as the kernza market develops. So, to summarize what we've learned so far, to help reduce agriculturally associated greenhouse gas emissions, we can utilize perennials over annuals and leverage the yield benefits from intercropping. To help reduce emissions and pollution associated with nitrogen fertilizer production and use, we can utilize legumes and intercrop to provide biologically fixed nitrogen to the system. To help reduce grain yield decline in kernza, intercropping with legumes should be investigated. And to help kernza grain farmers make up for small kernza grain yields compared to wheat, they can manage their system for dual use. Now that we see the why of the system, we can finally get into the how of our experimental objectives, designs, and analyses. In my first chapter, we investigated whether intercropping kernza with alfalfa can reduce the need for synthetic nitrogen fertilization while maintaining or improving grain and forage yields and quality compared to monoculture. We also sought to understand how yields changed over time in this intercropping system and with different levels of nitrogen fertilizer, as well as how all of these parameters varied based on planting locations across the United States. In my second chapter, we wanted to understand how alfalfa variety affects the performance of this intercropping system and compared alfalfa varieties with 3 different fall dormancy ratings. So, in order to understand the effect of environment on the Kern's alfalfa system, we replicated this experiment at 4 locations across the United States, here at Cornell, University of Wisconsin-Madison, University of Minnesota, and at the Land Institute in Salina, Kansas. We had 4 replicates at each location and 23 treatments in each prep. Treatments included 2 cropping schemes, intercropped and monoculture. Our monoculture plots were seeded at about 11.2 kilograms per hectare. Intercrop plots were seeded at 5.6 kilos per hectare of each species in what was known as a replacement intercropping design, which is where rows are replaced with the second crop rather than the second crop being planted in between rows, as we can see here. Kansas itself did have a slightly different planting scheme due to planting equipment limitations, but those differences were accounted for in the analysis. Data collection included kernza grain yield, kernza forage biomass, and alfalfa forage biomass. We also collected weed biomass, but did not include it in the following analyses. We harvested both grain and forage biomass in a quadrant basis by hand, and here we can see a hand harvested plot from last summer in New York. We completed summer harvests of grain and forage once grain reached maturity, and another harvest in the fall before first frost. In year 2, locations with available labor also harvested biomass before kernza stem elongation in the spring, but spring harvest data was not included in the following analyses. After the spring harvest, plots were mowed and urea was applied. Here we can see a picture of alfalfa biomass being harvested from a quadrat in the fall. The Kansas location did not have sufficient regrowth for any fall harvests, so we don't have their data for that. And we also don't have any year 2 data for Minnesota, as they had to replant their experiment in the next year due to rodent issues. Each replicate had both monoculture and intercrop plots fertilized at 1 of 5 different nitrogen rates, increasing in increments of 40 kilograms per hectare from 0 to 160 kilograms of nitrogen per hectare. We used 3 different alfalfa varieties, High-Just 360 with a fall dormancy rating of 3, Shockwave BR with a fall dormancy rating of 4, and WL365HQ with a fall dormancy rating of 5. Only Shockwave was planted at all 5 nitrogen rates because we didn't want to have hundreds of plots at each location. So High-Just and WL were only planted at 0 and 80 kilograms of nitrogen per hectare. So we have one big experimental design for both chapters, but use different subsets of the data for analysis in each chapter. Speaking of analysis, after the grain and biomass was harvested, forage biomass was ground and analyzed using near-infrared spectroscopy. In our modeling of forage quality, we use both crude protein values and relative feed values because they both indicate the nutritive quality and usability in livestock. The relative feed value is calculated using parameters from that near-infrared spectroscopy to help understand the performance of forage once fed to livestock. We analyzed the forage quality of each species separately, so we also calculated a total crude protein and total relative feed value for intercrop plots by adjusting the forage quality values based on the proportion of biomass yield made up by each species. And despite breeding efforts, kernza still has some crushability problems, meaning that the grain does not always easily separate from its outer hull, thus skewing some of the masses that we got from our grain yield. Here we can see some kernza grain still in its hull, and here is some dehulled. So we use digital imaging and analysis software to estimate the proportion of dehulled grain in a given subsample, which we then use to perform adjustments on the total grain yield per plot. We were unable to complete this imaging yet for year 2, so we used year 1 adjustments in order to calculate the year 2 dehulled grain yields. So please keep in mind that technically the year 2 grain yield results are preliminary moving forward. Forage harvests from each quadrat were converted to a kilogram per hectare yield, and then both mass-based yields and land equivalent ratios were modeled for biomass harvests. More on land equivalent ratios in the next slide. Linear mixed effects regression models were created for each individual harvest because we did expect to see large differences between both fall and summer harvests and year 1 and year 2. Results were log transformed to meet assumptions, and post hoc analyses included analysis of variance and pairwise comparisons. The factors that we included in the nitrogen rate study were, of course, nitrogen, environment, and cropping system, and factors included in the variety study were genotype, environment, and cropping system. So, as mentioned, land equivalent ratio. Our intercropping was in a replacement design, meaning that there's half the seeding rate of each crop in intercrop compared to monoculture. To be able to compare the performance between monoculture and intercrops, we can use the land equivalent ratio. To visually explain this, if here in yellow we have a monoculture of Kernza, and here in green we have a monoculture of alfalfa covering the same area, and here we see our alfalfa Kernza intercrop over the same area with half the total area taken up by each species, we, for example, may expect to see 6 kilos from a hectare of Kernza. That is not an accurate number, but easy numbers for example. And we may expect to see 4 kilos from a hectare of alfalfa. So with half the seeding rate in our intercrop, we'd expect to see half those yields of intercrop. But due to the facilitative effects of intercropping that lead to overyielding, as discussed earlier, we often actually see greater yields in a given land area. Like in this example, if we saw 3 kilos from alfalfa and 4 kilos from Kernza. Using these expected and actual yields, we can calculate the partial land equivalent ratio for each species and then sum them to find the land equivalent ratio. So in this example, the partial LER for Kernza would be the yield in intercrop compared to the yield in monoculture. And if we see a value greater than 0.5 for the partial LER, we then see an overyielding effect in that species. The same calculation can be done for alfalfa, comparing intercrop to monoculture, and if we see a value greater than 0.5, we see overyielding. And we can add these values together to figure out our land equivalent ratio for the entire intercrop plot and comparing expected or overyielding to expected. And if we see a value greater than 1, then we see overyielding in the whole intercrop plot. So with that, we can finally get into some results for the nitrogen rate study. We'll start with grain yield. So for both years, we saw significant interactions between environment, which is E in the table, and nitrogen rate, which is N in the table, as well as environment and cropping system, which is C in the table. Our stars indicate significance levels from our ANOVAs and at a p-value of 0.5, with more stars indicating a greater degree of significance. Here we can see mean grain yield values in kilograms per hectare at each nitrogen rate at each site, with lines fitted from our linear mixed effects regressions. Lines in red are from year 1 data, and lines in blue are from year 2 data, with dotted lines being monoculture Kernza and solid lines being intercropped data. As we can see in the graphs, nitrogen rate only had a significant effect on grain yield in New York in year 1 and at Wisconsin in year 2. For the sites that did not have a significant difference in grain yield between nitrogen rates, this indicates that applying nitrogen does not meaningfully increase grain yields in either cropping system, so a farmer could reduce or eliminate nitrogen application while still maintaining grain yields. With the environment by cropping system interaction, the effective cropping system did vary by site, but we only see a significant difference in grain yields between cropping systems in New York in year 1. No significant difference in grain yields between cropping systems shows that despite half the seeding rate of Kernza in intercrop grain yields were still comparable to monoculture grain yields. Overall, the effects of environment that we do see here and will see across many of our response variables may come from a lot of sources, including variability in temperature, precipitation, soil nutrient loads, soil types, and weed pressure, just to name a few. For example, here in New York, we had weed pressure in the form of white clover, which encroached on monoculture Kernza plots as the clover filled in the vacant legume niche. This could have affected the nitrogen load in the soil, although we did still see an effect of nitrogen rate in these plots in New York. Another factor that may have affected grain yields was lodging. We did observe lodging at high nitrogen rates for both intercrop and monoculture, and as we can see here in this picture of monoculture Kernza at 160 kilograms of nitrogen per hectare, lodging may not only decrease grain yield, but also make the grain harvest a lot harder for farmers who plan to use a combine to collect grain. Our grain partial land equivalent ratio results support these claims about the effect of cropping system and nitrogen rate on grain yield. We did not see significant effect of nitrogen rate on grain partial LER in either year, indicating that any overyielding effects are independent of nitrogen application. These graphs show the response of grain partial LER to nitrogen rate in both years at all sites. The dashed red line is at 0.5, above which indicates a yield advantage in intercrop. Year 1 and Year 2 grain partial LER results showed that grain yields had a yield advantage in intercrop compared to monoculture across nation rates and locations, except at New York in Year 1, Kansas in Year 2, and at 160 kilograms of nitrogen per hectare in Year 1 if confidence intervals are taken into account. We do see some outliers at Kansas, which may have skewed the Year 1 data in favor of a grain yield advantage in intercrop. But as Year 2 grain yields are preliminary, we do hesitate to make any strong claims about the effect of nitrogen rate or cropping system on grain yields over time. We generally do observe that Year 2 grain partial LER values were higher across locations and nitrogen rates in Year 2 than Year 1, which can indicate that intercropping may help ameliorate grain yields declining over time. Next, we can look at the forage side of this dual use system. There was a three-way nitrogen rate environment cropping system interaction in Year 1 summer, Year 1 fall, and Year 2 summer, as well as a significant effect of environment, as well as a nitrogen rate by cropping system interaction in Year 2 fall, which highlights that total forage yield reaction to cropping system and nitrogen rate is very site-specific, which does make sense as forage yields can be highly dependent on site-specific and annually variable factors like precipitation, temperature, and nutrient availability. Now would also be a good time to recognize that the alfalfa stands were not managed in a typical harvest pattern. Usually alfalfa stands are harvested multiple times over a summer growing season, which maximizes forage yields and quality. But because our kernza forage cannot be harvested in the summer to avoid disrupting grain development, alfalfa also cannot be harvested in intercrop. So we also do not harvest monoculture alfalfa until grain yield time as well. In this picture, you can see our senior research associate, Julie Hansen, holding up some of this late summer monoculture alfalfa. You can see it's very stemmy, it's lost a lot of its leaves, and therefore is going to have a pretty low forage quality. But because we're most interested in comparing intercrop plots to monoculture kernza, just to see how the presence of alfalfa benefits overall yield and quality, we still included monoculture alfalfa as a benchmark. That being said, here we have some graphs of our total forage biomass yields by nitrogen rate, cropping system, and location. We now have 3 lines comparing intercrop to monoculture kernza and monoculture alfalfa, with the different colors and representing different harvests across the 2 years. We only saw a significant response of nitrogen rate in monoculture kernza, where there was a positive correlation between nitrogen rate and total forage yield, which is expected for a grass that cannot fix its own nitrogen. We also saw in New York in Year 1, in their fall harvest, there was an inverse relationship between nitrogen rate and total forage yield in intercrop plots. Because we do not see a significant positive response to nitrogen in intercrop plots, this indicates that intercropping kernza with alfalfa provides the opportunity to reduce or eliminate nitrogen fertilizer application and costs while still maintaining high total forage yields when compared to monoculture kernza. Additionally, we found that intercrop total forage yield was significantly higher in monoculture kernza across nitrogen rates in the fall harvest of both years. We did not see this effect in summer harvest, but some locations did have higher summer total forage yields at lower nitrogen rates than monoculture kernza. A higher total forage yield in intercrop during fall harvests, but not summer harvests, shows that a system like this may be best for grain farmers looking to get higher yields and qualities from their fall forages, but that still recognize that their intercrop summer forage yields may not necessarily be higher than kernza monoculture. Across cropping systems, we generally saw a decrease between years 1 and 2 summer harvest total forage as expected. However, intercrop total forage yield was higher than monoculture kernza total forage yield for Year 2 summer harvest, indicating that the farmers could benefit from a greater total forage yield over time with the intercrop than monoculture kernza. We saw the most dramatic changes in total forage yield over time in Kansas across all cropping systems. In Year 1, we did observe a much higher total monthly precipitation at Kansas in the late spring compared to Year 2. So our hypothesis is that this precipitation may have increased soil water availability during either critical growth periods or just throughout the entire growing season, which could bolster forage yields and actually make the Year 1 forage yields more of the outlier than Year 2. We can look at our LER results again to understand whether intercropping conferred a total forage yield advantage compared to monoculture. We only see a significant response to nitrogen rate in year 2 summer, where there was an inverse relationship between nitrogen and LER, indicating that forage yield advantage in intercrop is more likely at lower nitrogen rates. In other words, a higher nitrogen fertilizer application in monoculture is more likely to match any yield advantage conferred by intercropping than a low nitrogen rate. Overall, LER results consistently showed an advantage for intercropping across nitrogen rates, except at 120 to 160 kilograms of nitrogen per hectare in year 1 summer and 160 kilograms in year 2 summer. We saw that LER values were higher in year 2 than in year 1 for both summer and fall harvest in Wisconsin, for summer harvest in New York, and at low nitrogen rates in Kansas. A greater yield advantage in intercrop in year 2 is likely tied with the fact that root and nodule decomposition is the main mechanism for nitrogen transfer from legumes, and these tissues just have had more time to turn over by year 2. So while forage biomass yields are important, forage quality is extremely important when feeding forage to livestock and therefore can have a large impact on profitability. We saw a significant three-way interaction in year 1 summer and varying interactions between the factors for other harvests. While our ANOVA results tell us that nitrogen rate had a significant effect on total crude protein, we in fact see that there was no significant difference in total crude protein between nitrogen rates within a given cropping system and location. Monoculture alfalfa total crude protein was significantly higher than monoculture Kernza, which is expected when comparing a legume to a grass. Total forage crude protein values in intercrop plots were higher than monoculture Kernza, regardless of nitrogen rate. This indicates that intercropping Kernza with the legume does in fact confer a forage quality benefit. However, the inverse relationship between total crude protein and nitrogen rate in New York summer harvest that we can see here was contrary to expectations that providing more nitrogen to the system would reduce nitrogen scarcity and therefore increase total crude protein. One hypothesis is that this may have been due to higher nitrogen availability, leading to a greater nitrogen allocation to the grain rather than to the forage, but as we don't have grain quality data, this hypothesis cannot yet be tested. Overall, both the general lack of effect of nitrogen on the total crude protein, as well as that decrease in total crude protein with increased nitrogen, indicates that farmers intercropping Kernza and alfalfa could again save money while maintaining forage quality by decreasing their synthetic nitrogen applications. We also observed that fall harvest had higher crude protein than summer harvest, which was expected with a shorter regrowth period between summer and fall harvest compared to spring harvest and summer harvest. While not statistically compared, we also generally saw that year 2 crude protein values were equal to or greater than year 1 values, which is expected again with transferring more nitrogen over time as tissues decompose. All right, so for total relative feed value, we only saw a three-way interaction in year 2 summer, but nitrogen rate only had a significant effect on total RFV in intercrop plots in Kansas in year 2 summer, where total RFV decreased with increasing nitrogen rate. Here we see that. Additionally, at New York and Wisconsin in year 2 summer, there was no significant differences in total RFV across cropping systems and nitrogen rates. This, with the fact that we did not see a significant effect of nitrogen during other harvests, indicates that there is no total RFV advantage to applying nitrogen to intercropped Kernza alfalfa systems. While we saw significant cropping system effects across harvests, intercrop total RFV was generally greater than or equal to RFV in monoculture Kernza across locations and harvests. Both total CP and total RFV were generally higher in fall harvest and summer harvest across locations, cropping systems, and nitrogen rates. Though we did not yet analyze spring harvests, we would expect to find similar things with spring quality higher than summer quality. This higher quality forage in these marginal seasons could be more valuable to farmers by helping bridge any existing feed quality gaps after the main forage growing season. So to recap our results from the nitrogen rate study, we did not see a significant effect of nitrogen on grain, total forage yields, or total forage quality in intercrop. Intercropping significantly increased fall total forage yields, and intercropping increased forage quality when compared to monoculture Kernza. And while not yet modeled, we generally observed that grain yields did decline over time, but preliminary results indicate that intercropping may decrease the rate of grain yield decline over time. These results indicate that a dual-use intercropped Kernza alfalfa system may be a viable route for maintaining Kernza grain production for more profitable systems for farmers when compared to monoculture Kernza, by reducing nitrogen input needs, and at those lower nitrogen rates, increasing grain and forage yield over time over a given area, increasing forage quality, and potentially maintaining higher grain yields over time. However, to truly identify the best system, further work must be done to understand the costs and benefits of the Kernza alfalfa system based on the cost of nitrogen and the value of the grain and forage yields over time based on the markets most readily available to farmers. Hopefully you're still with me. We have just a few more slides of results for the alfalfa variety study, which are luckily pretty straightforward. So first of all, even though we only fertilized all three of the varieties at 2 nitrogen rates, we found that there was no significant interaction between nitrogen rate and variety for any of the response variables. So we removed nitrogen as a factor in all models. There were also no significant genotype by cropping system, genotype by environment, or three-way interactions for any of our response variables, which indicates that no one variety is better suited to a single environment than another, nor are any varieties better suited to intercrop than another. We did see significant environment by cropping system interactions, indicating that intercropping success does vary with environment, which is what we also saw in the last chapter. So we'll start with our forage and grain yield results. I promise this slide is not as scary as it looks. We did not include Kernza monoculture yields in total forage quality or models because there is no level of variety of alfalfa from Kernza monoculture. So for Kernza yields, we only looked at variety performance in intercrop. That being said, we did see an effect of variety on alfalfa forage yields and total forage yields in year 1. For both alfalfa yield and total forage yield, the variety Shockwave BR led to significantly higher summer yield than WL365HQ and significantly higher fall yield than in High-Jest. This indicates that out of the 3 varieties, Shockwave BR may be the most profitable choice for forage yields, at least in year 1. Its performance may have been correlated with its fall dormancy rating, but it also may have had additional attributes not quantified in the study that may have led to higher total forage yields. While there's no significant differences in alfalfa or total forage yield in year 2, additional data in year 3 may better elucidate the trend in total forage yield changes over time. With the environment by cropping system interactions, intercrop total forage yields were higher than monoculture across sites and harvests. We did not see any effect of genotype on Kernza forage or grain yields, which indicates that no one variety increases Kernza yields in intercrop more than another. We did not see a significant effect of genotype on total LER across harvests, indicating that no one variety leads to a greater forage over yielding effect in intercrop compared to the other varieties. While no variety was statistically greater than another, all the varieties' total LER confidence intervals did exceed 1, indicating a re-yielding effect, except for Shockwave BR in year 1 summer, which is interesting considering we did see it had higher total forage yields. Like in the nitrogen rate study, we did see a significant effect on environment in total LER, as we can see reflected in these box plots. And we might not see it in these graphs because they're on different scales, but we did see a total LER that was greater in fall than in summer harvests. So we see similar trends in grain partial LER. There were no significant effects of genotype across years, which indicates that no one variety leads to a higher grain yield advantage in intercrop than another. While no variety was statistically greater than another, confidence intervals indicated that a grain yield advantage in intercropping is likely for both Shockwave BR and High-Jest 360 in year 1, but not for WL365HQ. Variety performance in intercrop, again, varies with environment. And we saw in year 1 in New York, confidence intervals actually did not show that intercropping gave a grain yield advantage for any variety, which may have been related to the lodging we discussed previously. So for our forage quality, there was no significant effect of genotype on total crude protein or total relative feed value, which indicates that no one variety confers forage quality advantage in intercrop over another. While the effect was not uniform across environments, there's generally a lower total crude protein and total relative feed value in intercrop compared to monoculture alfalfa, which is expected with a lower proportion of legumes in their crop. While we did not include monoculture Kernza in the analysis, we would expect there to be a lower forage quality than intercrop, as seen in the previous chapter. As well, we would also expect to see no effect on Kernza forage quality from genotype. To summarize our findings for the alfalfa variety study, out of the 3 varieties tested, we did not find one to be better suited to the environments tested, and we did not find one to be best suited to intercrop Kernza. We did find that variety had a significant effect on alfalfa forage yield and total forage yield in year 1, with Shockwave BR more consistently providing a higher yield than the other 2 varieties. Variety did not have a significant effect on Kernza grain yield or forage yield, total forage or grain over yielding, or forage quality. While we saw some effect of variety on total forage yields, we cannot definitively say that this was an effect of fall dormancy rating of Shockwave, as this was a rather narrow study, only using 3 varieties of 3 fall dormancy ratings, which may not have actually been representative of their fall dormancy group. We also only tested these varieties across 4 locations, and there may have been other attributes of the varieties besides fall dormancy rating that contributed to the role in these results. To bring together all of our findings from this Kernza alfalfa intercropping experiment, our results show that a farmer focused on Kernza grain yield production could potentially benefit from a dual-use Kernza alfalfa intercropping system with grain yield advantages during the summer and a higher forage biomass of higher quality in the fall, while increasing profits through synthetic nitrogen need reduction. We also found that fall dormancy rating may not have an effect on alfalfa variety performance in intercrop with Kernza beyond year 1 forage yields. Despite these initially promising findings, we recognize that the study was limited. We only have 2 years of data, only 4 locations, and only 3 alfalfa varieties with only 3 fall dormancy ratings. There's plenty of work this study and future studies could continue to do to overcome these limitations to better improve our understanding of the dual-use Kernza alfalfa system. In the study, we plan to include a 3rd year of harvest data next year, and longer-term studies are generally important for perennial systems. Both this study and future studies could be improved through more thorough modeling of relevant parameters, including the effect of pre-existing soil nutrient conditions and their changes over time, how the system and its management affects grain quality, and how confounding factors like weed pressure, row spacing, and interannual weather variability affects outcomes. Breeding of Kernza is ongoing and will continue to be important as we pursue larger grain sizes and yields, slower yield declines, and optimize Kernza for use in intercrop. And of course, there's plenty of other management effects that could be investigated beyond nitrogen fertilization. Different alfalfa varieties should be tested for or bred for intercrop. More work on that coming soon. And overall, while findings are promising, economic analyses will be very important to understand how reducing nitrogen fertilization and intercropping in a dual-use system actually affects profitability and farmer bottom lines. With that, I'd like to thank my community members for the guidance and kindness, and my collaborators for the patience and support. I'd like to thank my lab members for all the laughs in the field and all the help in the field, and of course, my friends and family for their unwavering love and support. It was such a privilege to work on this project, and I really love my time at Cornell, but I'm also very excited for my next chapter back in Virginia. And with that, thank you all for your time, and please feel free to ask me questions.
About the Kernza lodging, did you see an environment effect on Kernza lodging?
So we did not analyze that, but we did largely observe it mostly at New York in year 2. Other locations definitely have observed it. And there is some work saying that lodging rate is likely associated with higher nitrogen rates, but no one has specifically looked at environment effect of that yet. Yeah. But we'd expect to see that probably. Yes.
A very naive question about these systems. I know they're going to have to be renovated, like alfalfa has to be renovated. How long do these go before typically you replow and you renovate the whole system?
Right, so that's one of the problems with the Kernza forage yield decline. So in my first graph, we saw that those intermediate wheatgrass successions, they declined after 1 year. Typically, the literature says that the yield decline is after 2 years. And for forage yield decline, we typically see them after around 3 years for both Kernza and alfalfa. So right now we're looking at farmers probably wanting to renovate after 3 years, but the more work we can do on the system to keep yields higher for longer, we're going to have that greater perennial benefit over time.
So the typical alfalfa renovation would then be your cycle because the Kernza would keep the yield.
Yeah. Yes.
Good job, Megan. Thanks. I have a question about the grain of the plant. Why do you think intercropping would be?
That is the question. A large part of it is thought that by keeping nitrogen rates consistent and higher over time in the field, then we reduce that limitation and make sure that grain yields get to stay high. And also it might just be one of those intercropping effects that have not been fully understood yet. I think there's still more work that needs to be done there to truly understand the effects of that.
Yeah, I think Jake's work found that increased nitrogen was the opposite effect of grading decline, so like it helps the first year higher nitrogen rates make it worse. So if you do see an effect, really cool thing.
Yes.
And competition.
Yep, totally. Very cool. My fellow Kernza homie. Yeah. Yes. What's that? Hey.
Not my area, so sorry, the question didn't make sense, but my understanding of what you're saying is you're not seeing an effect of nitrogen rate on much of it, not for the food protein, not, I may look at it grain, but not on the yield effects. Where's all the nitrogen going? You put on all these different amounts, and what kind of cost, any ideas that I just might be. Like what part of the system is going into clean, is it running off?
That is a great question. So the question is about if we're not seeing a nitrogen effect, where is that nitrogen going in the system? Is it running off? Is it going to weeds? Those are both options. So when we're not seeing a nitrogen effect in intercrop, it is essentially because the legume is helping provide enough nitrogen that the Kernza does not need to continue to forage for the excess nitrogen. So we would expect it to either stay in the system or go to weeds or run off, or potentially if there are any other changes in nutrient balances, maybe go into other organisms within the system.
Yeah. So you could maybe work in soil tensile, so it'd be interesting to see what soil.
Yes, no, I totally agree. So we did some baseline testing on our soils, but it was not in depth. I'm not sure if all locations actually tested. And that was mostly just to make sure that our nitrogen and our potassium and our sulfur and our, what am I forgetting? You know, the big ones, yes, phosphorus, thank you. Making sure that they were all in balance. But I think that more in-depth analysis of nitrogen cycling throughout the whole system would be really interesting to understand where everything is going. Yeah. Sarah.
Oh, okay, that's my good. How do you harvest the farming system and how does that work on that?
Yeah, good question. So we hand harvested. Let me see if I can find a good example of what we did. Here. So you see how I'm basically just grabbing a bundle of Kernza. We basically just cut the grain heads off the bottom, we dry them, we brush them, but that's hard mode, that is experimental mode. Theoretically, we can use combines. One of our locations, Kansas, actually did use a combine to harvest in year 2 because they had limited data or limited labor for hand harvesting. So, and we do have some farmers that are actually growing Kernza on farm and they use a combine for that.
Yeah, thank you very much, Megan. That was great. A lot of work and a lot of intriguing data work that you do. I was wondering whether you had nitrogen uptake data and you could calculate from the trade-off between adding nitrogen and the lack of nitrogen uptake or somewhat a nitrogen transfer equivalence from legume to non-legume to answer maybe a question or provide some fine points on the mechanisms why nitrogen doesn't increase yield because it's a trade-off between getting it from the legume and getting fertilizer, and somewhat cleverly use the data to calculate that transfer rate.
Yeah, so we actually did calculate a parameter that we called relative yield, which actually means a lot of different things, but the way we calculated it, we compared intercropped plots at zero nitrogen rate to monoculture plots of Kernza and alfalfa at the different nitrogen rate levels. and using that proportion, then we could basically estimate how the intercrop plots with no nitrogen fertilizer compare to fertilizer effects in other plots. So we kind of did do some fun data stuff to approximate that, but we didn't really find any results that were surprising or exciting or necessarily worth including in the 45 minutes, but I do have that in my paper if you would like to see that data sometime. Yeah. But I agree that I think a more thorough understanding of where the nitrogen is going in the system is really important.
I had two questions. One is, do you know the yield decline that you get in the second year? Has anybody looked at nitrogen immobilization by the microbes? Are you sort of feeding the microbes instead of feeding the crop when you're adding that fertilizer? And then a follow-up question to that is, how much has the yield decline been investigated in out years? So if you waited 3, 4, 5 years to your yield start coming back because something is changing below ground, or they basically just stay low?
For your first question, we have not specifically investigated that yet in the system, seeing whether mobilization is a large factor in that decline. And in terms of whether or not we see regression, basically, I have not seen any of that in the literature. It pretty typically is just a steady downward decline. And there's a lot of thought on what may be causing that. Some of it is greater amounts of leafing out and tillering, well, not tillering, but basically reallocation of resources away from grain and towards biomass. Yes, Janice. Yes, good question. The question for people going here was whether or not we actually checked if the alfalfa had any nodulation going on. So we did use pre-treated seed for the alfalfa, so we hopefully saw some rhizobial association, but we did not check nodulation rates. So we were just assuming that the rhizomes were doing their job down there. And with the results that we saw at low nitrogen rates in intercropping, we assumed that they were giving some nitrogen. But with the fact that intercropping itself, even without legumes in the system, can still contribute over yielding effects, then it is hard to untangle that. So we did not check nodules, but we do assume that there were at least some doing their job. Yeah. Mm-hmm. If not, yes.
I have what you showed a slide with the cover in culture. So how much do we control on the side, and if that has impacted your fields across the sides, across the years, in terms of nitrogen application, was it done at one time or split application? How was it done?
Yeah, so nitrogen application was done in the spring, basically during regrowth and right after spring mowing, so around April, early May timeframe, and all the nitrogen was applied just at that one point in the year. And we could potentially see different effects if we did a split application throughout the year, but that's not the experimental design we went with. In terms of weed control, we largely left that up to site. So we know that, for example, Minnesota, I think, did some spraying for shepherd's purse, and Kansas had plenty of problems. They had alfalfa weevils, they had spraying for different yeah, so each site only did their management as they saw fit. So like in New York, we didn't really do much management, and we certainly saw plenty of clover. We see a lot of perennial weeds in the system, as people know. In perennial systems, you have a shift towards perennial weeds. We had lots of timothy, we have Canadian thistle, all that. And even our reps varied in how weedy it was. But yeah, so weed management is one of the large problems in a perennial system. You can't just, especially with a system that is both, that has grasses in it, you can't, you have different modes of action when you're spraying. You can't necessarily, just blanket apply any weed treatments. Yeah, thanks all for coming.
This has been a production of Cornell University, on the web at cornell.edu.